Right, for our next talk, we have Leandro, who's going to talk to us about new discoveries in the Brazilian Twilight Zone. Over to you, Leandro.
All right. Can you guys see my screen?
Yeah, that looks perfect.
Nice. So, hello, everyone. Thank you for being here today, and thank you for the opportunity to be presenting JETZON. So just a very quick introduction. My name is Leandro. I am Brazilian. I have been developing most of my career in Brazil, but currently I'm working in France, in Marbec, in the south of France. And the idea today is to present some of the work that we have been doing in the Twilight Zone of Brazil, but also more than that, show an example on how international cooperation can work in developing nations to improve the science in the Twilight Zone and in the deep ocean in general. So hopefully you guys will enjoy that. So let's see. All right, so we all know the importance of the Twilight Zone, so I will not be giving a lot of time on that, but just to give a very small context, we know that in the Twilight Zone, at least for most of the parts in the world, we have these huge biomass, incredible biodiversity, many ecosystem processes that are crucial for the functioning of the world ecosystems. However, we have this very huge lack of knowledge in many aspects of the Twilight Zone, but also in many areas in the world when we speak about the Twilight Zone. And we also know that we have these crescent threats that are now already changing the Twilight Zone. And given that many people around the world are developing amazing projects to study the Twilight Zone, and in JETZON we have very good examples. However, in developing nations are facing much more problems to keep up and to do this kind of research. So basically what's happening now is that the least known areas in the world is within the least developed economies. So we clearly have a asymmetry in the Twilight research. So in this context, the international cooperation is not a very good example on how we can correct this asymmetry, but maybe is the only way that we will truly achieve the goals of the decade of the deep ocean science, which is the science that we need for the ocean that we want. So the idea of this talk today, as I said, is to present what we have been doing in Brazil. So maybe we can improve our collaborations, but also show how we are doing this international cooperation and maybe also inspire you to be part of this partnership or any other partnership like that. So our research group, we call that Tapioca, which basically means Tropical Atlantic Interdisciplinary Laboratory on Physical, Biochemical, and Ecological Human Dynamics. We develop research in many ecosystems, but one of the main ecosystems, it is the Twilight Zone. So I will begin presenting our campaign, so what we have been doing there. So the first project is Abraços. Abraços in Portuguese mean hugs. So the idea was that different areas of the marine sciences hug each other to improve the twilight science in Brazil. So we could combine more than 100 researchers to work in this project. So there are people working with taxonomy, ecology, contamination, acoustics, chemistry, physics. So all the analysis that we could do in a multidisciplinary survey, we try to do in Abraços, so we could collect a lot of specimens and a lot of information, as I will be showing to you guys later. But the first campaign was in 2015, and the second one in 2017 in the northwest of Brazil. We have the coastal area, but also some oceanic islands that are very unique in this area. So our second campaign was in the end of the last year. The name of this project is Amazomix. The idea of this project is try to understand what is the influence of the largest river in the world into the Twilight Zone. So for instance, we know that the Amazon plume carries a lot of nutrients, organic matter, and also a lot of contaminants. And we know that there is a huge influence in the Atlantic. So we want to evaluate what is the influence of this plume in the twilight zone. So for instance, we know that a lot of mesopelagic species are maybe relying on the organic matter from their main source of energy. So we want to establish the connection of a river input into the trophic ecology, biomass, and biodiversity of mesopelagic species. So here are a couple examples. So with these two projects, we were able to collect a lot of specimens, and for each one of these specimens, we try to collect as much as possible information. So here is an example on how this kind of cooperation can work. Just to mention, the main comparison that we are doing is between France and Brazil. So mainly with the IRD, which is a French institution that support research in the South Ocean. So here is some example of the information that we are trying to collect for each one of the species. So we are doing work on taxonomy, functional biodiversity, different kinds of contamination, DNA, autolysis, bioluminescence, vertical migration, acoustics, anything that we can do with our information we are trying to do. We have different groups of people working with different aspects, but at the end, we always try to combine all of this information. And with this data, we are able to answer several questions. Here, I will give some examples mainly for fish, because that was the subject of my PhD. So I will be speaking a little bit more about fishes, but we also have all of these questions for crustaceans, cephalopods, jellyfishes, and other kind of taxonomic groups. So just to give an idea of what kind of questions we are doing, we began with simple questions, but simple but important, such as what are the mesopelagic fish species? And we didn't know that because we didn't have mesopelagic cruises in this part of Brazil. So what are the main group of species? How these species are distributed three-dimensionally? How is the relationship with physical oceanographic conditions? What are their main prey and trophic relationship, and what are their functional roles? And also, what are their main threats now and in the in the following years? So beginning with the diversity of fishes, we collected much more species than what we were expecting. So that was really nice and surprising. So far, we were able to identify 201 truly mesopelagic fishes. 50 of these species were new to the Western tropical Atlantic, and so far 8 are new to science, and we are currently describing. And also we could see through our analysis that if we kept the sampling in the same area with the same gear, we would have been collecting much more species. So it means that if probably we go back then, there we will find much more species, which is really nice.
Sorry, Leandro, sorry to interrupt. I'm not sure your slide is presenting properly in that we can still see your research questions.
Oh,
that's
now,
better. That's better.
right.
I can see it now.
Nice. Thank you, Mark.
That's all. Thank you.
So this is the main groups of species that I was explaining. So totally was 201 species and 8 new to science, and we could also establish the main group of species, which has some similarities with other places of the world, but also some particular cases that we could only see in Brazil. So we just recently published an article summarizing all the biodiversity of fishes in the area. But also for each one of the main group, we have been doing specific articles dealing with the taxonomic information on the new species and stuff like that. I will not go in details because of the time, but if you want to check out, I will leave here for you guys look later, or you can send me an email. But after we address the taxonomic questions, we move for more questions about ecology. But here are an example of some of the species that we have collected. They are just incredible animals. So here are a couple examples for the fishes, but also, as I said, we have crustaceans, cephalopods, jellyfish. So for each one of these specific group, we had a lot of new information that now we are trying to make it available. So after the diversity and the taxonomy, we move for questions more related to the ecology of these species. So here are a couple examples for the main species of fishes. We tried to combine with physical oceanographic conditions to see which one of the species are doing vertical migration, what's the relationship with the thermocline and the minimal oxygen layer, which species can go to the very hot waters of the epipelagic zone in Brazil. So that was really interesting to do because we saw a lot of different patterns. So not all the species have the same behavior. So it means that they have different functional roles, for instance, in the transport of carbon. So we could establish that. When we address the trophic ecology using stable isotopes and gut content analysis, we also could see some interesting patterns. So for instance, we could see that gelatinous spray it seems to be much more important for mesopelagic species, at least in our study areas, than we thought. And we couldn't see that before because we were using just gut content. But now that we are including a lot of stable isotopes information of jellyfish, we can see that these species likely have a trophic role much higher than we were expecting. And also we could combine all of this information to provide some overviews of the functional roles of these species and also their vertical niche partitioning. So here are examples of 2 articles that we published summarizing that. So for instance, here we can see that even species that morphologically are very similar, they can have a very different behavior to avoid competition. So for instance, here in the lanternfishes, when the species are in the same depth layer, they seem to be not sharing the same trophic pattern. And when they share the same trophic pattern, they do different kinds of migration. So it's a very complex way of doing vertical niche partitioning. And also in the headset fishes, which is the second most abundant fish group in our samples, we also could see a similar pattern where the species seems to have this very complex way of niche partitioning, which of course reflect in their functional roles. So we are looking the niche partitioning, comparing different species like that, but also comparing the twilight zone species with other ecosystems. So for instance, here we use stable isotopes for a lot of species from seabirds to the deep sea. We are calculating the niche of these species and try to see what is the similarities and the difference from the twilight zone. And for instance, we can see that in the epipelagic zone, we have much more overlapping of trophic patterns. However, the deeper we go, the lesser is the niche partitioning of these species, which means that the twilight zone species have this very complex way of diminishing the competition, and maybe that is why they have this diversity that is so huge and incredible. We are also looking, comparing the epipelagic and twilight zone in different ways. So here in this, it's a work that we are currently developing. But for instance, through the stable isotopes, we can see very clear differences between the mesopelagic and epipelagic species. We were expecting difference because the microbial looping changed the isotopes, but we were not expecting so strong differences. So which means one of our hypotheses is that in the very warm and oligotrophic waters of Brazil, where the vertical migration can be a little bit difficult for some species, the twilight zone species are relying more on the organic matter, and that's why we can see a much more clear difference between the mesopelagic and epipelagic zone than in other places of the world. This is a work that we are still developing, and hopefully this will be published soon. But we are very excited to use stable isotopes again from seabirds to the end of the twilight zone to see what are the difference and the dissimilarities between these species. Here is another example, not only a scientific example, but the importance of the partnership. So here is an overview that we did for the viperfish. It's a very emblematic mesopelagic species. And basically what we saw is that in Brazil, these species were not caught in the epipelagic waters, which again was a little bit surprising because usually in other parts of the ocean, we could see the viperfish in epipelagic waters. So we decided to use our data and combine with all the information available for the viperfish, and we calculated a model of the suitable habitat of the viperfish. And we can see here in the first picture in blue is the suitable habitat of the viperfish according to the temperature. And basically what we can see is that in the temperate zone, these species can go to the epipelagic waters because the waters are cold. But here in the tropical zone, this species remains into the deep sea all the time because of the very strong thermocline and the hot waters in the epipelagic zone. So we combine all the information available that we could find for the viperfish to calculate this model. And basically what we could conclude here is that the ecology and also the functional roles of the viperfish can be modulated by the latitudinal change in temperature. So for instance, in the tropical zone, there is no strong relationship between the viperfish and the epipelagic predators such as tunas, but that's not true when we go to temperate zone, where the viperfish, for instance, can be an important prey for tuna and dolphin fishing and other species of predators like that. So of course, this is a study case for only one species, but it is likely that many other species follow the same behavior. So when we try to calculate the importance of fishes in the transport of carbon and questions like that, we indeed have to consider some local differences when we speak about temperate and tropical zones. And this study was interesting to show some aspects on that. So as I said, we are also doing some work in contamination. So far, we have been mostly addressing microplastic contamination. So for instance, we could show that species that are relying on organic matter are more likely to be contaminated by microplastic. But now we are also moving to other kinds of contaminants, especially in the Amazon plume, such as heavy metals and organic contaminants. So I believe that soon we will have also some information available for that. And basically, that's it. I already included our survey in JETZON, so we would be very happy to help and contribute to the group. And just to close, we are expecting to have much more new information with our last samples from the Amazon region. We are expecting to do new analysis, new questions, and of course, new collaborations. So we are open to that if anyone is interested to work in the Brazilian waters as well. So I hope that with this presentation, I could inspire you to be part of this kind of collaboration, if not with us, if not with Brazil, with any other nation that would benefit of this kind of collaboration. And I hope that we always remember that we are not separated by the ocean, but in fact, we are connected through the ocean. And only working together, we will correct this asymmetry in the Twilight science and really achieve the goals of the decade of deep ocean science. So thank you very much. Feel to contact me if you want, and I hope that you have enjoyed that.
Excellent. Thank you very much, Leandro. Huge amounts in there, and a nice sentiment to end on as well. Do people have questions or comments for Leandro? So Leandro, I've got one. So you've got a huge amount of data, so it's going to keep you busy for a while. And so you were showing the sort of the niche structure within your data. Have you got a feel for how diversity varies between niches? I mean, do you find that some niches have a greater diversity of organisms than others?
Well, now we are also doing some work on functional diversity of fishes mostly. And what we can see, at least for fishes, is that the deeper we go, the higher is the diversity. So we believe that is related with niche partitioning. But more than that, so far we didn't look at that. But for sure, what we are looking at, we can very clearly see now, is that the deeper we go, the higher is the differences in the niches of these species. They have like small niches and very separated niches.
That's interesting. I mean, when you say small niches, I mean, how small? I mean, are they sort of very finely tuned for sort of quite small ranges of whether it's temperature or oxygen or whatever it is?
Yes, it's more like the niche of prey,
right.
But also they are restricting the prey that they choose. What is very kind of surprising, because usually we have this thought that in the deep sea the species will eat wherever they find, but actually they can be very predictive in the prey that they will eat. And so we can see that, but also we can see that some species are more in specific layers. So we also believe that this is a way of increasing the niche partitioning. So we can see very clear peak of abundances for specific species in specific depths.
Interesting. Very complicated, obviously, but it's interesting to hear that there are potentially such strong connections between species in terms of, you know, predators are a little more selective of what they eat than you might think. Because I have to admit, I'd assumed that, you know, when you get down into the twilight zone, you eat what you bump into, provided you can fit it in your mouth. But obviously, it's a bit more selective than that.
Yes, exactly.
Right, any more questions or comments for Leandro? Let me just check in case we've got anything on the chat.
I do have one. So you had like a whole diagram where you were looking at the different how do you call it, like research you guys want to do, like looking at plastic, looking at bioluminescence and all these research, but especially for bioluminescence, have you done any, or have you found any significant findings in these fishes that are living in the twilight zone?
Well, that's a good question. Actually, the work on bioluminescence, we are just beginning now to collect mostly bacteria that is related with bioluminescence. So we are carrying on the analysis like at this moment. So no finds so far, but hopefully in a couple months we will have more information on that.
Okay. You've got too much data. That is your problem.
Yeah.
And it's interesting. I mean, having the Amazon as sort of sort of feeding into the twilight zone. I mean that's just a huge field in itself. Extraordinary. Any more questions or comments for Leandro? I see we're starting to come up to our hour, but we've definitely got time for another question if anybody's got one. No, I don't see anything. So once again, I'm sure that Leandro would be delighted to hear from you if you have any questions or comments after the talk. So thank you very much to Nestor, Nicolina and Leandro once again for their talks. Thank you all for joining us on this call. And as I said, we'll be advertising the next call before long. But otherwise, thank you very much.